First speaker of this session is me. I am me. I am from Georgia Tech. I work co-advise between Nick Head and Lauren Williams. We're really lucky at Georgia Tech to have a lot of funding for Origins of Life research. So quick plug. So I'm working in the Center for Ribosomal Origins and Evolution. So that's why today I'm going to be talking about what's probably going to drive some of the rock people in here crazy, which is molecular paleontology. So in this case, we're kind of working on a top-down approach where we're trying to use what's left in modern biology and drill back as far as we can into evolutionary history with that. And it turns out, luckily, we can drill back pretty far. So you guys are already generally familiar with the ribosome. You're about to become intimately familiar with the ribosome. So you guys have already seen the central dogma. This is where the ribosome fits in here. So the ribosome is what turns RNA into protein. It is, in terms of biomolecules, huge. It's 1 megadalton, 1 million daltons. It is, as I already mentioned earlier, probably well established, maybe not in its full modern form, but in a pretty advanced form at the time of the last universal common ancestor. This is generally how it works in a sort of schematic way where it reads the messenger RNA, and these tRNAs serve as adapter molecules that turn that code into the specific amino acid that code codes for, and then you make a protein out of that. This is what it looks like in more depth. So here we have separated out the small subunit, which is responsible for binding the messenger RNA. And it is comprised of what's called the 16S ribosomal RNA and about 20 ribosomal proteins. But I don't care about that this morning. I'm going to talk to you solely about the large subunit, which is responsible for the peptide bond formation. So it's pretty widely accepted that the small subunit came later in evolution. So we're trying to go back as far as we can, so we're just going to look at the large subunit. So it's where the actual chemistry happens. So the 23S and 5S ribosomal RNAs are the RNA components of the large subunit. Again, I'm not going to talk about the 5S because it's much later. We're going to talk solely about the 23S RNA and a subset of these 30 proteins. So when you're looking at this here's the 5S RNA. I will generally tell you today that if something looks like it's on the exterior, it's probably newer. So the 5S RNA you can see is right out here on the outside, probably a lot newer addition to the ribosome. And then these beige things here that look like helices, those are the 23S and the 16S ribosomal RNAs. And these purple guys that are sort of stuck in all over the surface, those are the ribosomal proteins. So this in particular is a bacterial ribosome. Eukaryotic ribosomes are even bigger. So what we wanted to do was try and resurrect an ancestral version of the ribosome. So what we called it was the Ancestral Peptidyltransferase Center, APTC for short, and basically because that's what we wanted to do. We wanted to be able to make peptide bonds, to do the peptidyltransferase reaction, but in a much more simplified way than we have today. So included in our model are the ancestral ribosomal RNA. It's a very stripped down version of the 23S RNA, which I'll show you on the next slide. And it's stitched together with these tetraloops, which all you need to know about those is that they're very stable. We know that they fold in a very predictable way. So they're kind of anywhere that we made a cut in the RNA, we put those in. And 5 ribosomal peptides that are highly conserved and penetrate very deeply down into the core of the ribosome. Those are derived from ribosomal proteins, large subunit ribosomal proteins L2, L3, L4, L15, and L22. All you really need to know about those is that there's a bunch of them, this is just a few of them, and that the L stands for large subunit. So in this case, in the background here, you can see the entire 23S ribosomal RNA for comparison, so you can see how much we really have stripped away in this. Really, it's about 80% of the RNA, and so these are the co-crystallized ligands from this crystal structure that just give you an idea of where in this beast the actual chemistry happens. So it's right down in here, right in the center. So this is what the secondary structure of the 23S ribosomal RNA looks like, secondary structure, rather than this here, which is a 3D structure, is basically just laid flat for simplicity's sake. So anywhere that there's a sort of two lines close to each other, that means that there's base pairing going between those two strands of the RNA. So in the dashed line here in the back, that's the entire 23S ribosomal RNA. In the black and the sort of blue is what we kept. So you can see again, we'd stripped a lot of this away. We actually made this part in the lab, it represents about 20% of a bacterial large subunit RNA. And made it with some nice PCR techniques that I wish I had the time to tell you about, but I don't. And these ribosome peptides, which I can tell you about more in a couple slides. But first I need to tell you about my methods. Main method that I use is called SHAPE, and that stands, actually Jessica showed some of this yesterday, but it stands for Selective 2' Hydroxyl Acylation Analyzed by Primer Extension. Astrobiologists and their acronyms. So the way that it works in a very basic way is you have a chemical that selectively modifies your RNA at positions where it is flexible, i.e. not base paired. So what you get in the end is a readout of the places in your RNA that are not base paired. And then from there we do reverse transcription into just using an enzyme. We hijack biology basically. To make the complementary DNA that stops when it hits one of these modifications. So what we're left with is a library of pieces of DNA that represent the places that are flexible in the corresponding RNA. We run that on capillary electrophoresis, which if you know anything about gel electrophoresis, is exactly the same, but it's run in a capillary. From there, we can process that data. Actually, we use in-house MATLAB scripts that we've developed to get a single nucleotide readout of the flexibility of that piece of RNA. So we did that on this ancestral piece of ribosomal RNA because we wanted to see basically if, regardless of the fact that we stripped away about 80% of the RNA, if it still folds in a similar way. And it does. What you can see here is that anywhere that there's these triangles, that means that it is flexible, and those triangles line up really well with the single-stranded regions, which we expect to be flexible. So we were very happy about that. And in this case, I want to highlight that this was done just with the RNA on its own and some monovalent cations, which just help to neutralize the backbone so that you get secondary structure formation. You get the helices actually able to form. So no divalent cations or none of the ribosomal peptides. We did have some hints early on that certain parts of the secondary structure that we were working with were not great. This is going to be a little bit of an aside, but for instance, this region here, I would have expected these nucleotides to be reactive, but they were not. But when I went back and looked at the actual 3D crystal structure, it looked a little bit more like this, where my data lines up a lot better, and I was happy again. So we had hints early on that the 23S secondary structure, which we derived this representation from, was not perfect. And so some guys in our lab decided to take that and run with it and rewrite the ribosome Bible, essentially. So that big secondary structure I showed you at the beginning is largely the way that it looks because of historical purposes. And as young scientists, we all know how much we hate that sort of stuff. So in this case, they basically redrew the map so that it didn't have a gap between the two halves for starters, and so that it represented more accurately the base pairing that's seen in the crystal structure. So in particular, it's all just brought together, and this domain here, which we called domain 0 because they'd already named the others 1 through 6, a little facetious maybe, but really makes up the core of the ribosome. So what you're seeing here in blue is the ancestral ribosomal RNA just overlaid on this new secondary structure. So going back to the ribosomal proteins / peptides, this is what the ribosomal proteins in general look like. They have these big globular domains and these long fingers that have sort of really no structure that penetrate down deeply into the ribosome. They vary in size depending on how far they penetrate and how much of it actually penetrates, from 18 amino acids to 45. This is what they look like if we look at the crystal structure of the ancestral ribosomal RNA with these individual peptides. So you can see that they're kind of peppered around. In some cases, they get pretty close to the very middle, which is where the chemistry happens. But for the most part, what these are here for is to stabilize the backbone so that you can get these negative charged pieces of RNA coming closely together. So previously, we've been able to show that 4 of those 5 ribosomal peptides do actually bind our ribosomal RNA. I don't have time to go into this, but if you'd like to discuss it later, I'd be happy to. This is a bunch of experiments that I didn't do, but the only one that didn't was L2. But we didn't know if it was binding in the exact place that we wanted it to. So that's where my research came in, and I went through and used the crystal structure to say, hey, where these things actually, or where do we want them to bind? And in the secondary structure, it looks something like this. Just in the different colors are the different locations that we might expect to see contacts if they're binding at the expected places. This is what my raw shape data looks like. I'm not going to try and help you guys interpret it too much, except to know that anywhere, so any location with an arrow here that you kind of see a peak that increases or decreases. So there's some that appear as we increase the amount of these peptides. In this case, it's the L4 peptide. That means that there is a change in the structure at that location. It's a local, in a lot of cases, just a single nucleotide that's maybe becoming less flexible, so maybe more lockdown or becoming more flexible and floppy. So far I've done L4 and L22 peptides, and when I compare where those actual changes happen, this is in a very rough way. I haven't fully processed this data yet because it's kind of a nightmare to process. But if I sort of do a really rough estimation, these are the positions that I see some of the biggest changes at. And so for the L4 in particular, let's say this is one of the places that I do see a change, and that is one of the places that I expect to, down here, that I expect to see a change. So very close, which makes me very happy. And again, right here, there's some not too far away that I expected to have a change in their structures. So what I can say right now is that both the L4 and L22 peptides do induce modest changes in the structure, and in some cases at locations near where we expected them to. And L3 alone I didn't show because it's boring. It doesn't seem to bind, at least in a specific way. So it may still bind, but just not the exact way that we intended. It may be that it's dependent on having one of those other peptides there first. So that you need to have one so that the other one can kind of cooperatively come in and bind. And in the future, we're going to start doing these in combination so that we can get an idea if there's an actual order of assembly and that sort of thing, and also start adding back in stuff like magnesium, which is also very important in forming large RNA structures. And that's everything that I had. These are all the people that helped with my work. And once again, thank you, NASA, for funding us and for funding this conference in a large part. And also the Center for Ribosomal Origins and Evolution. I will happily take any questions. Bradley? I see. You have a question. This better be good.
Oh, it's awesome. All right. So you mentioned your future studies doing magnesium, adding it there.
Yes.
Can you see where I'm going? Any temptation to try it with iron, since I know your labs have the capabilities for that?
Yes. Yeah, that's definitely a down the road sort of thing too. Yeah, because iron has been shown to really mimic magnesium in terms of the way that it interacts with RNA. So yes, for certain, I would like to do that. Part of it is just instrumentation issues. We are in the process of getting an anaerobic chamber that will allow us to do it, but we got it from somebody else who cut it in half and tried to put it back together, and so it leaks. So we're getting the manufacturers to fix it. But once we have that, we should be able to do all kinds more interesting stuff in terms of like anaerobic iron 2 chemistry.
Okay, cool. That should be fun.
Mm-hmm.
Other questions?
Okay. Thank you very much.